A very good morning all of you. Today we will discuss an important pathway in biochemistry, that is heme synthesis. You can remember this pathway by remembering this mnemonic, that is some great doctors palpate heart under cover and produces pure heme. So this is the mnemonic to remember the steps in the heme synthesis. We'll discuss the steps one by one. Before that, the site for the heme synthesis, it is erythroid precursor cells. Near about 80% of heme synthesis occurs in erythroid precursor cells and remaining occur in the liver. Then it is mitochondria and cytosol both. The first enzyme, that is ALA synthase, it is present in the mitochondria, and all oxidases, that is coproporphyrinogen oxidase, protoporphyrinogen oxidase and ferrochelatase, they are present in the mitochondria. So it is partly cytosolic pathway and partly mitochondrial pathway. Then we will discuss the steps in the heme synthesis. There are 4 important steps. The first one is the synthesis of parphobilinogen. Second step, the conversion of parphobilinogen into the uroparphyrinogen. Then third, conversion of this uroparphyrinogen into protoporphyrin. And fourth one, incorporation of heme of iron in the protoporphyrin. These are the 4 important steps in the hemoglobin synthesis. Now we will discuss one by one step in the heme synthesis. Now this is the mnemonic, that is sum grade. The sum, it is succinyl coenzyme A, an intermediate in the TCA cycle, condenses with the glycine. Glycine is an amino acid. In presence of enzyme, the enzyme in this step is the ALA synthase. It gives del aminolevulinic acid. Del aminolevulinic acid. It is written as del aminolevulinic acid. This reaction is PLP dependent. Pyridoxal phosphate is required in this reaction. It is required to activate the glycine. Then in this reaction there is removal of coenzyme A, there is removal of sulfhydryl group, and also there is decarboxylation. There are 2 reactions in this. Succinyl coenzyme A plus glycine gives alpha amino beta ketoadipate, and then this intermediate is on decarboxylation is converted into del aminolevulinic acid. This step is catalyzed by the ALA synthase. So this is the some great doctors. Now coming to the next step, in which 2 molecules of del aminolevulinic acid condenses to form a parphobilinogen. Parphobilinogen. Now this is a monopyrrole. There is formation of pyrrole ring. and the enzyme required in this step, it is ALA dehydratase. ALA dehydratase. There is dehydration, there is removal of two water molecules. Two molecules of del-amino-levulinic acid condenses with the removal of H2O. Two molecules of H2O gives parphobilinogen. Here is the formation of pyrrole ring, or it is considered as monopyrrole. This one is the zinc containing enzyme and it is inhibited by the lead. This enzyme contains zinc. This one is the zinc containing enzyme and it is inhibited by lead. And deficiency of these two enzymes, ALA dehydratase and ALA synthase, leads to the anemia. Now coming to the parphobilinogen, which is a monopyrrole, there is formation of one pyrrole ring. There is, in the next step, 4 molecules of parphobilinogen condenses with the removal of ammonia. It gives a compound, the name of compound is hydroxymethylbilane. Hydroxymethylbilane. This one is a linear tetrapyrrole, means there is formation of pyrrole ring in the first step, that is parphobilinogen. In the next step, there is formation of linear tetrapyrrole on deamination. This is connected by the methylene bridges. The enzyme in this step, conversion of parphobilinogen to hydroxymethylbilane, the important enzyme is uroparpyrinogen 1 synthase. This one is the important enzyme, and deficiency of this enzyme lead to the disorder known as acute intermittent porphyria. The another name for this enzyme is HMB synthase, that is hydroxymethylbilane synthase or parphobilinogen deaminase. There are 3 names of these enzymes. This is important in the entrance purpose. They are deficient in the acute intermittent porphyria. The names are hydroxymethylbilane synthase or parphobilinogen deaminase or uroparpyrinogen 1 synthase. Deficiency of this enzyme lead to the disorder known as acute intermittent porphyria, which is characterized by the abdominal pain and neuropsychiatric manifestations. Okay, so this is some great doctors palpate heart. Now, in the next step, this hydroxymethylbilane undergoes cyclization and there is formation of uroparpyrinogen 3. There is formation of uroparpyrinogen 3 with the help of the enzyme, that is uroparpyrinogen 3 co-synthase. And deficiency of this enzyme lead to the disorder known as congenital erythropoietic porphyria, and all porphyrias are dominant except congenital erythropoietic porphyria. Okay, there is cyclization. This is linear tetrapyrrole, this is monopyrrole. Here is the formation of single pyrrole followed by the condensation single pyrrole which get converted into linear tetrapyrrole with the help of 4 parphobilinogen molecule on deamination. Then on cyclization it gives uroparpyrinogen 3. Okay, this is the structure of uroparpyrinogen 3. These are the pyrrole rings. This is the pyrrole ring, and it has 8 substituent groups, that is AP, 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 and PA. As it is PA here, there is change in the sequence AP, 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 and PA. That's why this is known as 3 in the series. That's why the name is given uroporphyrinogen 3. If it is AP, 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 and again AP, then it is uroporphyrinogen 1. So this is about uroporphyrinogen 3 synthesis. After this step, this uroporphyrinogen 3 it get converted into coproporphyrinogen 3. Uroporphyrinogen 3, it is converted into coproporphyrinogen 3. And the important enzyme in this reaction is uroporphyrinogen decarboxylase. Because decarboxylation is there is removal of 4CO2 leads to the synthesis of coproporphyrinogen 3. What happens in this step? This is the structure we already discussed. This is the structure of uroporphyrinogen 3, that is AP, 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 and PA. On decarboxylation, there is removal of 4CO2 molecule. This convert this acetyl group into the methyl group. This acetyl group is converted into methyl group. Now MP, 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 PM. So this is coproporphyrinogen 3 with the removal of 4-CO2 with the help of enzyme uroporphyrinogen decarboxylase. This is also an important enzyme. Deficiency of this enzyme lead to the disorder known as porphyria cutanea tarda, and this one is a most common porphyria cutanea tarda with cutaneous manifestation, that is photosensitivity. This photosensitivity is mainly due to the release of lysosomal enzymes hydrolases, and that causes itching and photosensitivity on all. Now, with the help of next enzyme, that is coproporphyrinogen oxidase. As it is oxidase, it is present in the mitochondria. So onward enzyme, that is coproporphyrinogen oxidase, then next enzyme and next enzyme, these last three steps, they occurs in the mitochondria. Here also there is removal of CO2 molecule, and due to the removal of CO2 molecule, what happens? The propionyl group, which is present at the first two positions, means this is the structure. There is MP, 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 PM. This propionyl group, it is substituted by the vinyl group, and that is, it is converted into MV, MV, MP, and PM. And this is known as protoporphyrinogen 9, or you can name it protoporphyrinogen 3. 3 and 9, they are same in the porphyrins. So this protoporphyrinogen 9, with the help of, again undergoes oxidation with the help of protoporphyrinogen oxidase. It get converted into protoporphyrin. and this after this formation of protoporphyrin, there is incorporation of iron in the ferrous form. This protoporphyrin, the iron gets center position in the pyrrole ring and it gets converted into heme. Here is the formation of heme, and the important enzyme in this step is ferrochelatase. The another name for ferrochelatase is heme synthase. So this is all about heme synthesis. We'll discuss one by one, that is condensation of succinyl coenzyme A, which is an intermediate in the TCA cycle, combined with a neutral amino acid glycine with the help of pyridoxal phosphate dependent enzyme ALA synthase gives del amino levulinic acid. Two molecules of del amino levulinic acid condenses with the help of enzyme, that is zinc containing enzyme ALA dehydratase, which get inhibited by the lead and gives parfobilinogen. Four molecule of parfobilinogen condenses to form hydroxymethylbilin, which is a linear tetrapyrrole. After this hydroxymethylbilin, it get converted into uroparphyrinogen 3. This uroparphyrinogen 3 is converted into coproparphyrinogen 3, which gets converted into protoparphyrinogen 9, and which is converted into protoporphyrin, and iron get central position and it get converted into heme. This one is the condensation reaction with the removal of CO2, coenzyme A and sulfidyl group. This one is the removal of water molecule is here, dehydration is there. Then there is deamination with the help of parfobilinogen deaminase or uroparphyrinogen 1 synthase or hydroxymethylbilin synthase. The three names of the same enzyme. Here this linear tetrapyrrole undergoes cyclization with the help of co-synthase enzyme and gives uroparphyrinogen 3, which on decarboxylation means acetyl group is converted into methyl group, gives coproparphyrinogen 3. Then propionyl groups, which are present on the first and second position, they get converted into vinyl group and we get protoporphyrin 9. And this protoporphyrin 9 is converted into protoporphyrin, which is converted into heme. The iron gets central position and it is converted into heme. So remember the mnemonic: some great doctors palpate heart under cover, produces pure heme. Okay, we'll discuss all or we will summarize the entire pathway in one diagram. We'll discuss the porphyria in next lecture in detail. Okay, this is succinyl coenzyme A plus glycine. It gives del amino levulinic acid. Condensation of del amino levulinic acid gives parfobilinogen. Four molecules of parfobilinogen combined to give hydroxymethylbilin. Then it gets converted into uroporphyrinogen 3. It gets converted into coproporphyrinogen 3. Then it gets converted into protoporphyrinogen 9. It is converted into protoporphyrin and it gets converted into heme. Okay. Now the important enzyme in this is the ALA synthase. There are two types of ALA synthase. One is present in the erythroid cell, erythroid precursor cell. The synthesis of heme occurs in all cells except matured RBC. ALA synthase 1 and ALA synthase 2 is there. ALA synthase 1, it is present in the erythroid precursor cell. It is a inducible enzyme. Various drug induces the concentration of ALA synthase and heme synthesis. Also, it is a rate limiting and regulatory step in this pathway, and it is regulated by the feedback inhibition and also by the repression and control of transport across the mitochondria. That is ALA transport across the mitochondria is also regulated. So this one is the important enzyme ALA synthase. Then second one is the ALA dehydratase. These are the two important enzymes. Then U1, U3, UD. These are the enzymes, that is uroporphyrinogen 1 synthase, then uroporphyrinogen 3 co-synthase, uroporphyrinogen decarboxylase, then coproporphyrinogen oxidase, then protoporphyrinogen oxidase, and ferrochelatase. These are the important enzymes. That is uroporphyrinogen synthase. The another name is parphylinogen deaminase or hydroxymethylbilin synthase. Then uroporphyrinogen 3 co-synthase. Then uroporphyrinogen decarboxylase. Then coproporphyrinogen oxidase, protoporphyrinogen oxidase, and the last one is the ferrochelatase or heme synthase. So these are the important enzymes in the heme synthesis. 1, 2, 3, 4, 5, 6, 7, 8. Now coming to the important porphyrias, there are two types of porphyria, hepatic and erythroid porphyria. So this enzyme is inhibited by the lead. Deficiency of uroporphyrinogen synthase lead to the disorder known as acute intermittent porphyria. This one is the most common acute disorder characterized by abdominal pain and neuropsychiatric manifestations. Then uroporphyrinogen 3 co-synthase leads to the disorder known as congenital erythropoietic porphyria. This is the only porphyria which one is autosomal recessive, all are autosomal dominant. Then uroporphyrinogen decarboxylase lead to the porphyria cutanea tarda. This one is the most common porphyria and it having cutaneous manifestation, that is photosensitivity. Then coproporphyrinogen oxidase mitochondrial enzyme, it lead to the disorder known as hereditary. Then next one is the protoporphyrinogen oxidase. Deficiency of this enzyme lead to the disorder known as erythropoietic porphyria. The deficiency of this protoporphyrinogen oxidase leads to the disorder known as variegate porphyria. And ferrochelatase enzyme deficiency lead to the disorder known as protoporphyria. So these are the various porphyrias in which porphyrins and various intermediates are excreted in the urine. The acute intermittent porphyria, congenital erythropoietic porphyria, porphyria cutanea tarda, hereditary coproporphyria, variegate porphyria, and protoporphyria. This is all about the heme synthesis. We will discuss the porphyria in our next video lecture. Keep watching. If you like the video, please hit the like button and subscribe to the channel. Thank you all of you. Thank you very much.